Well, hello and welcome to the seventh lecture regarding cell-cell contact given by Professor Janusz Solushi. So, the thing about this lecture is that there are a lot of slides, and a lot of these slides don't give that much of a sense. Some of them are very vague, and some just give a lot of numbers and information. So what I'm trying to do is that I will try to make sense of the whole concept of this lecture, which is cell-cell and cell extracellular matrix contact, and I will talk about the material which I found important in the exams and by talking to the teachers and other students. So first of all, we need to make a distinction between two different things: multi-adhesive matrix proteins and the adhesive proteins and receptors which are located on the receptor. The first group, which are multi-adhesive matrix proteins, are components of the matrix, and you need to know three of them: laminin, nidogene, and fibronectin. You can see the structure of laminin and nidogene here and their molecular weight. Well, here the only thing you need to know, as far as I'm concerned, is that laminin and nidogene, they're both multi-adhesive matrix proteins, and that laminin is formed by three polypeptide chains. And that is all the things you need to know about these two. But about fibronectin, you need to know more. First of all, fibronectin is a heterodimer. As you can see here, there are two monomers forming the fibronectin, and these monomers are different. So it is a heterodimer. And the main function of fibronectin is anchoring cells to the extracellular matrix and contributing to the shape to holding the shape of cell and cytoskeleton. And these functions which I just mentioned are the main function of most of multi-adhesive matrix proteins. But the thing which is very important and quite unique about fibronectin is that it has got a role in migration and differentiation of cells, especially during embryogenetic development. So if fibronectin has got a role in migration and differentiation of cells, it's common sense that in case of tumor cells, in case of cells which are migrating and dividing without any regulation, these proteins are not present or are mutated. So tumor cells do not secrete these proteins. And one other thing about fibronectin is that fibronectin has got a RGD motif. Now, RGD is a three amino acid sequence, which is arginine, glycine, and aspartate. Now, what is the importance of having an RGD motif? That is for binding to integrin, because integrin, which is another type of protein which we're going to talk about just in a moment, has got binding sites specific for RGD, and macromolecules of the extracellular matrix, a lot of them, like fibronectin, they have got RGDs as a motif, so they can bind to integrins. Now, you can see here, if this is the three-dimensional presentation of the whole fibronectin, this part is the RGD. So this is an RGD presentation. And RGD, as I said, is arginine, glycine, and aspartic acid, or aspartate. Now let's talk about the other protein which we just mentioned, integrin. Integrin is also a heterodimer, and integrin mostly acts as a cell surface receptor. It can also act as a binding as a cell-to-cell contact or cell-to-ECM or extracellular matrix contact protein. Now, what makes integrin very interesting is that it has got a RGD specific binding site, which binds to proteins with RGD. So here you can see a heterodimer integrin, which is on one side, on the extracellular portion of it, it's binding to the fibronectin, and fibronectin is bound to a collagen fiber, which is one of the common components of extracellular matrix. So instead of collagen, it could be other components of extracellular matrix. And in case of instead of fibronectin, it could be another type of multi-adhesive matrix protein like laminin. And here we've got integrin, which connects this whole thing here to the components of cytoplasm of the cell, in a specific actin filaments of microfilaments, because as you probably know, actin filaments together form microfilaments in the cytoplasm. So this binding happens via some adapter proteins, and that is the importance of integrin. Now, one more thing about integrin is that it forms homophilic interactions. Now, what does a homophilic interaction mean? We are gonna talk about it right now. So you see, we've got two types of interactions between these receptors. They can either be homophilic or heterophilic, and here you can see that they can bind through extracellular linker molecule. But then again, even if they bind through these linker molecules, this can be a homophilic or heterophilic type of binding. Now, homophilic bindings means that these receptors tend to bind with themselves. Like, for example, if we've got a p-selectin, it wants to bind to another p-selectin, which is that was an example, because I don't think p-selectin is a homophilic protein, but that was an example. So if you've got an X receptor, it tends to bind to another X receptors on the membrane of other cells. But heterophilic binding means if you've got, for example, an X receptor, it tends to bind to the Y receptors on the other cells. So that is important. That can come in your exams. Now let's talk about all these adhesive proteins. We already talked a bit about integrins. We said integrins, they are heterodimers formed by two different they're formed by two different monomers, like fibronectin. Fibronectins are also heterodimers. We said the type of interaction between integrins are heterophilic. And here you can see that we are going to talk about four main adhesive proteins: cadherin, Ig superfamily, integrin, and selectin. And two of these are heterophilic and two other are homophilic toward other receptors. So among all these proteins, only Ig superfamily do not need calcium for performing their action. So all other three of these, integrin, selectin, and cadherin, they all need calcium ion in order to perform their action. So calcium concentration is needed for each of these proteins except Ig superfamily in order to work. Now let's talk about cadherin, which has got homophilic binding properties. And there are different types of cadherins: E-cadherin, N-cadherin, P-cadherin. I don't think you need to know where each of them is, but E-cadherin is important because we're going to talk about E-cadherins when we're talking about extravasation. So maybe just know that E-cadherins are present in the epithelial cells. And as we said, the cadherin molecules, in order to work, the cadherin proteins, in order to work, they need calcium. And other things that you need to know about cadherin is that they facilitate cell-cell contact in a specific. Now, you're gonna read about most of the material in this lecture, also in histology. And in histology, probably your teacher will say that cadherins are very important in case of binding of in case of cell-cell junctions in zonula occludens or adhesion belts. And then your teacher in histology will say that in case of desmosomes, which is another type of cell-cell junction, you're gonna have desmoglein and desmocolin instead of cadherin. So they make a distinction in histology between cadherins and desmoglein and desmocolin. But here in cell biology, desmoglein and desmocolin, which are present in the desmosomes, are considered to be a subtype of cadherins. So bear in mind that desmogleins and desmocolins are specific types of cadherins. And what cadherins do, as I mentioned, they are for cell-cell contact. So each cadherin protein on one side, on the extracellular portion, will bind to another cadherin protein, and on the other side, on the cytosolic portion, it will bind to the cytoskeleton of the cell. And that cytoskeleton of the cell can be either microfilaments or actin filaments or keratin intermediate filaments. Well, in case of cadherins themselves, they bind to microfilaments or actin filaments. But desmoglein and desmocolin, they bind to keratin intermediate filaments in the cytoplasm. So that is the big difference between cadherins and desmoglein and desmocolin. Now, let's again talk about Ig superfamily. Well, Ig superfamily consists of a big variety of proteins like NCAMs, which can be found in nerve cells. But in specific, we're going to talk about ICAM again when we're talking about extravasation. Now, again, as we mentioned, they do not need calcium for their action, and I do not think you need to know about sialic acid and about their alternative splicing. So now that we've talked about Ig proteins and cadherins, let's talk about selectins. Now, selectins, they also can be found in three main types: E-selectin, P-selectin, or L-selectin. They have several transmembrane domains, which you can't see in this picture, but they have several transmembrane domains. And they bind to carbohydrates. They have a lectin portion. These selectins, they have a lectin portion, which is important for calcium binding and interaction. So the lectin portion of selectins bind to these calcium ions, and the whole selectin then will bind to carbohydrate domains of glycoproteins or glycolipids. And because selectins tend to bind to carbohydrate domains which are different from selectins themselves, their type of interaction is considered to be heterophilic. And the other important thing about them is that they are also important for extravasation. Now, when we reach to extravasation, you already know about three of the things which we're going to talk about there: selectins, cadherins, and also I think we talked about selectins, cadherins, and integrins. Yes. So, what is extravasation and what exactly happens during extravasation? Well, extravasation is the process in which white blood cells or leukocytes will go from the blood vessels to tissue to perform their action, to perform defense mechanisms. And we are gonna now talk about how that process of extravasation occurs. Well, first of all, we'll have an injury, and at the site of injury, epithelial cells covering the lumen of blood vessels. So you can imagine that this cell here is an epithelial cell covering the blood vessel. So this leukocyte or white blood cell is moving in the blood vessel. This portion is the blood vessel lumen. This portion is the tissue which is injured. And now when the tissue is injured, there will be signaling mechanisms that will activate pathways in the cell, which will cause the extravasation. And I will define extravasation again. I'm sorry, which will activate the exocytosis, and I will define exocytosis again. Exocytosis is the process in which vesicles will fuse with the cell membrane and will extrude their content to outside the cell. So the exocytosis of some specific vesicles will be activated, and the content of these vesicles will be P-selectins. So P-selectins will be exocytosed to the lumen of blood vessel, but they will be still bound to the surface of the cell via some of their domains. And when they are on the surface of these epithelial cells, leukocytes, which have some specific carbohydrate domains on their membrane, which are glycoproteins or glycolipids, will bind to these P-selectins. These carbohydrate domains, these yellow ones, will bind to P-selectins. And since this binding is not that strong, the cell will not remain so long in this state. It will tap along other epithelial cells and other P-selectins until a specific type of factors are synthesized on the surface of epithelial cells. This black triangle here, PAF, or platelet activated factors. Now, when platelet activated factors are expressed on the surface of epithelial cells, platelet activated factor receptors or PAF receptors on the leukocytes, while leukocyte is tapping along these epithelial cells, these PAF receptors will find these PAFs and will bind to them. And when PAF receptors bind to PAF, a specific type of pathway will be activated in the cell, which will cause the phosphorylation, of cytosolic portion of integrins. Now, this reddish structure here is integrin. And integrins, as we mentioned, they have a cytosolic domain and an extracellular domain. Now, integrins are not always active. They need to be phosphorylated from the cytoplasm in the cytoplasmic portion in order to be activated. So this PAF receptor binding will cause a pathway which will result in the activation of these integrins. And when integrins are activated, they will bind to specific type of receptors on the epithelial cells, which are called ICAMs, which we talked about just a moment ago. And when that happens, everything is perfect for the cell to perform the extravasation and to go out and to reach the injured tissue and to perform its action. So now we're going to talk about the other part of this lecture, which is about the cell junctions. First, we're going to talk about the cell junctions on the lateral domain of the cells, as you can see here. And then we're going to talk about the cell junctions on the basal domain of the cells. So first, we're going to talk about tight junctions, adhesion belt, desmosomes, and gap junctions. And then we're going to talk about hemidesmosomes and focal adhesions. So as you can see, tight junctions here are the most apical type of junctions. It means they are closest to the apex of the cell. They're closest to this end of the cell. And just under tight junctions, there is the belt-like adhesion belts, and under the adhesion belt, there is the plaque-like desmosomes. So, let's talk a bit about tight junctions. Tight junctions they bind cells, they connect cells, especially epithelial cells, together on one side, and on the other side they are bound to actin filaments, and their main job, as written here, is to prevent the fusion of membrane compounds between apical and basolateral regions. So it means they form these very tight junctions that will keep cells very tight to each other, so the diffusion of material will be prevented. And these are tight junctions. They mostly work like fences, nothing more. But now we're gonna talk about adhesion belt. Adhesion belts or zonular adherents are a bit different. They are belt-like type of junctions, a belt-like zone on the cells, just under the tight junctions. And on one side, the cadherins of one cell, cell 1, will bind to the cadherins of another cell, cell 2. And in between, calcium ions will facilitate the interaction between these cadherins. So you can't see calcium ions here, but calcium ions are needed, are compulsory for interaction of these cadherin molecules. And you can see here, the type of these cadherins is E cadherin. Why? Because we're talking about epithelial cells right now. So on the extracellular portion, these E cadherins will bind to each other from different cells. Whereas in the cytosolic portion, cadherin molecules will bind to actin filaments or microfilaments, these pinkish structures, via some adapter molecules. A good example to memorize are catenins. So one side to each other cadherins will bind, on the other side they will bind to actin filaments via adapter proteins. And as we said, their action is also calcium dependent. Now let's talk about desmosomes. Well, desmosomes are very like zonula adherents or adhesion belt. The differences are the differences are cadherins in case of zonula adherents are attached to the actin filaments or intermediate filaments in the cytoplasm of the cells, whereas desmosomes, the specific type of cadherins in desmosomes are attached to keratin intermediate filaments in the cytoplasm of the cells. So they are attached to different types of cytoskeleton. That is a big difference. And again, the type of cadherins in case of desmosomes are called desmoglein and desmocollin. And I say it again, in histology, your teacher may distinguish completely between desmoglein, desmocollin, and cadherins. But in cell biology, desmoglein and desmocollin are a type of cadherins. Now, I think there is only one more lateral junction to talk about, and that is gap junctions. We already talked about gap junctions in another lecture, and we're going to talk about them again because the gap junctions are present in several lectures because they are important actually. Now, each gap junction is formed by two connexons. You see these structures here, these two structures, each of them is a connexon, and each connexon is formed by 6 connexins. These ones in a two-dimensional form. So again, these connexins in two-dimensional form will form one connexon here. As you can see, one connexon, and two connexons together, they will form a gap junction. And through gap junctions, all type of ions can pass. Actually, all type of charged particles can pass until their rate is less than 2000 Dalton. So there is only a size prohibition here. If something's size or rate is less than 2000 Dalton, then it will be able to pass through these gap junctions. And as we said in other lectures, these gap junctions contribute to formation of electrical synapses, because in case of electrical synapses we need free and rapid diffusion of ions for generation of an electrical impulse, and these gap junctions are just perfect for that purpose. And these gap junctions are regulated, their action is regulated via calcium concentration and pH. So calcium concentration and pH can regulate opening or closure of these gap junctions. Now let's talk about two last type of junctions, hemidesmosomes and focal contacts. Now the main difference here, as you can see here, is that the other types of junctions which we talked about, they attach cells to each other, whereas these junctions, hemidesmosomes and focal contacts, they attach cells to the ECM, to the extracellular matrix, or to be more specific, to basal lamina. And again, you're gonna learn about all of these again in histology. So let's now first talk about hemidesmosomes. Hemidesmosomes are very like desmosomes in the sense that they attach to the intermediate or keratin intermediate filaments in the cytoplasm of the cells, exactly like desmosomes, if you remember. And that sometimes you can see uh, same proteins working in both of them. But the difference is that we have got integrins in case of hemidesmosomes, which we didn't have in case of desmosomes. Now, integrins, I'm sure you remember, they are heterodimers and they have heterophilic interactions. And on the other side, as we said in the beginning of the lecture, in the extracellular matrix side, these integrins are bound to components of extracellular matrix in a specific multi-adhesive matrix proteins like laminin in this case, or fibronectin. And these multi-adhesive matrix proteins themselves are bound to other components of extracellular matrix, like in this case collagen. And that is for hemidesmosomes. Let's now talk about focal contact. Focal contacts are also like hemidesmosomes in the sense that they also use integrin molecules for their actions. But in case of what they attach in the cytoplasm, these focal contacts are very alike zonula adherens. Even some of the adapter proteins, which help the attachment of these focal contacts to the actin filaments or microfilaments, are same in both case of focal contact and zonula adherens or adhesion belt. And it's good to memorize the name of these adapter proteins, which are vinculin, taurine, and alpha-actinin. And again, in the extracellular portion, in the extracellular matrix portion, they do the same thing. They help the binding of components of extracellular matrix to the cytoskeleton of the cells. And that was all the cell junctions. So this lecture is over. I see you in the next one.